My name is Mariana Kiehl, and as a principal application scientist at Roche, I truly enjoy using my background in human genetics and oncology to help improve genomic technologies.
My name's Sandra Theall. I'm a scientist at Roche, and I'm passionate about next-gen sequencing and how it helps to move genomics forward. Both of us are based in the Wilmington, Massachusetts office, where we work on optimizing library prep workflows for a range of NGS sequencing applications. Between us, we've had extensive hands-on experience with many workflows, and that's why we're excited to talk to you today about our latest DNA library prep kit. One of the first things I noticed with the EvoPlus V2 workflow is how streamlined the workflow felt compared to the HyperPlus kit. When you open the kit box, there's just a few ready mix tubes that has everything you need ready for library prep. With fewer manual steps and reagents that tolerate vortex mixing, there's a lot less room for error, a lot less time pipetting, and greater consistency across runs. Another big advantage is that for labs looking to automate or scale up, the plate format integrates seamlessly with automated liquid handling systems. This saves time and reduces touch points, which is especially valuable for high-throughput studies.
As a scientist working with multiple applications, I really value the versatility that the KAPA EvoPlus V2 kit offers, especially through its library size tunability. For whole genome sequencing, for example, a narrow distribution of larger fragments is desired, while for hybrid capture-based sequencing, smaller inserts are performing better. With the KAPA EvoPlus V2 kit, library sizes can be adjusted at two different steps. First, the size of the DNA fragments can be optimized by changing the duration of the enzymatic fragmentation, with longer incubations leading to smaller fragments. Then second, if needed, a fine-tuning can be achieved with bead-based size selection. So unlike most of the transposase-based methods, the KAPA EvoPlus allows us to get the right sizes for the different applications and sequencing platforms.
After optimizing library size for your application, it's essential to make sure your libraries meet the expected size and concentration requirements. The EvoPlus kit makes this QC process simple and precise. Libraries can be quantified with fluorometric methods or qPCR-based assays with the KAPA library quantification kit. Then they can be run on a gel electrophoresis assay to check for library size and to detect any adapter dimers before sequencing. The EvoPlus kit provides the flexibility to incorporate the true library size into your qPCR molarity-based calculations, eliminating the uncertainty that comes with default size assumptions with transposase-based workflows. By verifying both library size and concentration prior to sequencing, you can confidently achieve optimal loading concentration for the sequencer, avoiding the issues that come with under or overloading, such as poor cluster density, uneven coverage, and reduced data quality.
All right, let's talk about enzymatic fragmentation artifacts. In the perfect world, the enzymes we use to fragment the DNA, they act like random biological scissors, but in reality, they don't. Enzymes like endonucleases and transposases, they have intrinsic sequence preferences, and this can lead to different types of artifacts or biases. One of them is the start site bias, which is defined as the uneven distribution of where the sequencing reads begin, and this can lead to non-uniform representation of a genome. In addition, artifactual variants, like unexpected SNVs, insertions, and deletions, can also be produced during enzymatic fragmentation. The enzymes in the KAPA EvoPlus V2 kit are developed to minimize all those artifacts, bringing it to levels as low as the ones observed in mechanically sheared samples prepared with the KAPA EvoPrep kit, for example. Minimizing these artifacts, it's all about data integrity. It ensures that your sequencing data really reflects the composition of your biological sample.
The EvoPlus kits, similar to EvoPrep, are designed to simplify your library prep workflow and adapt to your sequencing application. They deliver flexibility, reproducibility, and confidence from tunable library sizes and easy ready-mix reagents to seamless QC that ensures optimal loading and high-quality data.
For genomics scientists and other biomedical researchers, this kit provides a reliable way to create libraries that meet the criteria for different applications and sequencing platforms.